This presentation will aim to give an insight into the neuromodulation technique of transcranial direct current stimulation, or tDCS for short, and its efficacy in acting as a clinical treatment for major depressive disorder. We'll start with looking at the physiological mechanisms of major depressive disorder. Then we'll move on to the physiological effects of tDCS, then the effects of tDCS on major depressive disorder. Then we'll review and analyse some of the relevant literature in the field and make suggestions for future research. When attempting to understand the physiological effects of tDCS on major depressive disorder, it is first important to understand the underlying physiological mechanisms of major depressive disorder, or MDD for short. MDD is characterised by an inability to experience pleasure, hypersensitivity to negative stimuli, or low moods. Research has revealed that many factors can play a role in an individual's susceptibility to depression, including environmental stresses, lifestyle, and genetic comprise. However, on a neurobiological level, the brain areas that play a significant role in MDD are the amygdala, which is closely associated with emotions, the thalamus, which directs high-level functions like speech, thinking, and learning, and the hippocampus, which relates to memory. Researchers have also found that the following neurotransmitters play a role in depression. There's acetylcholine, serotonin, norepinephrine, dopamine, and GABA. And a better description of their functions are offered on the current slide. So what is tDCS? Transcranial direct current stimulation is a relatively non-invasive form of brain modulation practice, which uses a weak, direct current attached via two opposing scalp electrodes. These electrodes are placed over specific cortical areas of the brain in hopes of improving cognitive function. It is aimed at sending electrical impulses that affect the membranes of the neurons in order to create action potentials, ideally making long-lasting changes in cortical regions, targeting neuronal excitability, typically leading to an excitatory effect by means of neuronal depolarization. These two electrodes are named the anode and the cathode, and the current flows between them, influencing brain areas situated between the two of them. This has been confirmed by researchers through use of neuroimaging studies such as fMRIs. tDCS has been used therapeutically for neurorehabilitation, treatment of chronic pain, epilepsy, and neuropsychiatric disorders. Often the stimulation runs for about 20 to 30 minutes and the current intensity varies, but effects are expected to last long after the treatment is complete. As the left dorsolateral prefrontal cortex is the brain region responsible for suppressing the influence of negative emotional stimuli on behaviour, it is found to be hypoactive in the brains of people with MDD. So when administering tDCS for MDD treatment, a nodal stimulation to this region is believed to increase excitability in this area. This normalizes activity, hopefully resulting in effective influence to neurophysiological and behavioural outcomes. And so, opposite the anode, the cathode is placed in the lateral aspect of the right hemisphere. Some studies have reported not only successful polarization, but also strengthened synaptic connections, modulated neurotransmitter systems, and promoted synaptic plasticity and changes in resting membrane potential. The efficacy of tDCS has been contentious, with some meta-analyses showing that there is not enough reputable evidence to support the clinical utility of tDCS in MDD treatment. However, in their comprehensive review, Palm and colleagues considered a variety of meta-analyses, placebo-controlled trials, open-label trials, and case studies. They concluded that there was some efficacy in treatment of acute depression, and whilst effectiveness of tDCS decreases with resistance to treatment, the therapy still has very few side effects, so it should be considered. They deemed 20 minutes of 2 MA current strength per day to be a safe quantity. The typical critiques to literature of this nature still apply to transcranial direct current stimulation on major depressive disorder. And as it is a complex psychological disorder, limitations in control trials include possible dissimilarities to stimulation protocols, between patient variation in brain anatomy, and difference in patient selection criteria. Future studies should aim to include larger sample sizes with more representative samples to MDD and compare tDCS to other therapeutic neuromodulation techniques in order to eradicate these criticisms.